Yeah. Okay, let's start. So it's my pleasure to introduce Yina Liu to give the department seminar. Yina was an undergrad here in ESS before going off to do a PhD at Texas A&M, and she then did a postdoc at Hui first or at PNNL first? After
Hui first, and
Hui
then
first,
PNL later.
then PNNL to do a high resolution mass spectrometry, which I think she's going to be part of what she's going to talk about today. So it's my pleasure to have Yina back. Go ahead, Yina.
Thank you for the introduction, Francois, and thank you so much to have me. It's kind of a bit weird to be in this room. The last time I was here, I was one of undergrad attending a seminar. So you can imagine the feeling. So if I have stage fright, it's because I never given a seminar to a room full of people who I've taken classes from. So at any rate, today I'm here to talk about something I learned since grad school and then propagate to my postdoc and then back again as a faculty member to look at this very dear to my heart class of carbon called halogenated organic carbon and try to tell you what's going on about them in the environment using compound discovery. And I'll tell you soon what that really means. If I can advance the slide. Okay, so because you have two screens here, and I knew that, right? So I'll use this laser pointer so everybody can see that. So I'll anchor to this point. So what are organic carbon that contain halogen? Halogen organic carbon is that any organic molecule that contain at least one halogen atom, chlorine, bromine, or iodine. So historically, HOC, halogen organic carbon, are actually thought to be produced for and by humans primarily. So this panel of chemical structure here shows you some of the structure example of persistent organic pollutants. Some of them came from fire retardants or making solvents or electronic chip supplies and things like that. The reason to show you this structure, besides I'm obsessed with molecule structure, is to show you the diversity of the diversity of the structure, and note that many of them have aromatic rings. These compounds are thought to be, are known to be persistent in the environment because they're very energetically expensive to break down. The more halogen they contain, the harder to break them down in the environment. And then later, actually, in the natural product world, scientists started to find that actually there's classes of naturally produced HOC that if you compare the structure, they look quite similar, except that this type of organic molecule having halogens on them are naturally produced. Except that most of the effort to look at HOC in the natural sense have been focusing on natural product discovery because some of them have pharmaceutical value. So in the sense of identifying some of the natural products back in early 2000 was actually more than 5,000. So hopefully they're actually a bit more identified now. But then I'm coming in to ask this question. So if they can be naturally produced, what does that mean to the Earth system, or rather ecosystem, however we want to say that, Earth system, because this is the ESS department. So I proposed this schematic. It may look a bit complicated, but I'll unpack each one for you. So organic carbon in general in the ocean can be halogenated either through photolysis or enzymatic catalysis. Through algal or bacterial derived enzyme, or the exudate is organic carbon, or the carbon from the terrestrial environment that gets basically halogenated. And there could be two products that can be formed. When they are volatile, they actually contribute to the catalytic destruction of stratospheric ozone. However, I do have a reason to believe that there is actually another portion that are not volatile, and some of those can be pretty persistent, just how we think about the persistent organic pollutants. And this is a schematic of my salesman pitch, and hopefully this talk I'll convince you this is indeed happening in the ocean. So today's talk, I'll organize that into a little story of three pieces. Number one is to introduce the idea of mechanism to forming volatile organic HOC, and how they actually link to the non-volatile part of the HOC in the ocean. And then to show you a case study to show that they in fact actually were found in marine sediments. And then we asked another question of how long this can be preserved. And because I somehow have a collaborator who are great enough to share peatland samples that are actually can be dated back to 10,000 years ago, that become another great example to number one, show that some of this compound can be very stable. Number two is they are everywhere in the Earth system. So atmosphere, seawater, sediment, and peat, which is a terrestrial ecosystem. So during my PhD time, I focused on volatile halogen organic carbon, VHOC, for the reason that they are naturally produced. They also deplete stratospheric ozone. One of such example is bromoform, CHBr3. They are important for ozone destruction chemistry because they carry multiple bromines, and we know that bromine halogen is the key ingredient for catalytically degrade the ozone in the stratosphere. And because anthropogenic ozone depleting substances are being phased out, so it's actually now important to understand in the future climate, when you have a natural variant lows, what does it mean for the atmospheric chemistry? And also bromoform, 6-OBr, is what we call a very short-lived halogen organic compound, unlike methyl bromide. So where they get emitted become very important to the ozone depleting dynamic, because if they happen to be produced in places where you have deep convection that can just deliver them fast to the stratosphere, of course, then their efficiency to degrade ozone will be greater. So that actually was what's driving my dissertation research to identify the source of bromoform. So for decades, scientists actually thought that to be actually released by phytoplankton, and that's when I kind of went down to this rabbit hole, I guess, to try to identify who produced that. So I've been on cruises, except that this particular cruise I was not on because it happened in the 90s. My advisor, I don't know whether Eric went on the GasX 98 cruise, but this is GasX 98 data. So what I want to show you here is this red color data point shows bromoform concentration elevated compared to the cruise track mean. So they're higher than normal. And the gray bars or data points shows that they are not elevated. And then these points are actually superimposed on a ocean color chlorophyll concentration map. So what I want to draw your attention is in May of 98, the cruise was actually happening in Florida, I believe, and then they're going to Portugal. And what is interesting is the cruise that the ship was riding on this front of high chlorophyll region, and you can see elevated bromoform everywhere. This is leg 1. If you want to look at the actual absolute concentration, leg 1 is actually elevated compared to the similar location in a different time. Do I close caption? Do I need to, can I just close that, or someone will allow that? Okay. All right. So 1 month later, in June of 98, the ship was traveling back from Portugal, went into this zone of, again, high chlorophyll. And because it's also coastal environment, you can see elevated bromoform concentration. But what I want to draw your attention is leg 3 is slightly, I guess, offset from leg 1, but the key here is no longer riding on the high chlorophyll region, and we do not see elevated bromoform concentration. So leg 1 and 3, they have pretty stark difference in terms of concentration, then leads us, the scientists, to ask if we were to try to do a global emission model, what would be the best parameter to prescribe a emission field that would probably tells you that might be chlorophyll. So as an eager graduate student, then I volunteer myself to do chlorophyll concentration measurement and try to look at the correlation of the different cruises that conducted in Pacific and the Atlantic Ocean, and try to see, do we actually see a significant correlation between bromoform concentration in seawater and chlorophyll A concentration. For cruises that I participate in, I actually measure them. For those that I was not in, because it was before my time as a graduate student, I extracted satellite remote sensing data to try to glean some information of whether they're significantly correlated. As you may be able to tell from this cross plot between chlorophyll A concentration and bromoform, the conclusion will be they're actually not correlated. So if we try to use that as a model parameter, there might be some issues there. So then we can ask this question of exactly how something like bromoform is being produced in the presence of phytoplankton. If we look at some known halogenation mechanism, we know that there's a class of enzyme called the haloperoxidase. It could be actually from algae, phytoplankton, or from, if they're on land, it could be from fungal enzyme. On land, the fungal enzyme is actually have a role in degrading lignin. In the ocean, the role of haloperoxidase is actually rather peculiar. It could be actually try to scavenge superoxide when the cell was stressed, for example. So in the presence of superoxide and halide ions, halide use X to denote that, but that really means it's either chlorine, bromine, or iodine. With these two ingredients, it actually turned into something called a hyperhalolist or bromonist, or whatever halogen that you have as this formula XOX. If we think about that, when the X is chlorine, is in fact bleach. So another ecological reason to think about why do algae produce that, it could be a defensive mechanism to basically either fight off competition or other reasons. In macroalgae, was actually shown that if the macroalgae blade was actually wounded, they have more of this enzyme seem to be upregulated. So there's actually an ecological reason related to algae in the ocean. The interesting thing though is haloperoxidase is an extracellular enzyme. It could be freely released into the seawater or bind to the outside of the cell. So now, when they have bleach, we know from a life experience, of course, they react with organic carbon. It could actually have two main branch of outcomes. The not so interesting path will be there's nothing happen. But there's another pathway that might be very interesting, is that they produce non-volatile HOC. And through repeat halogenation of the non-volatile HOC, the larger molecule may start to cleave off and release the volatile HOC, such as bromoform. So if we then focus on some of this pathway, we start to understand that there is actually a connection with organic matter in the environment. There's a study by Stephen Manley. Actually, he did some of the research in Eric's lab, looking at the source of bromoform. And he found that if he removed DOM from seawater, it reduced bromoform production, again, highlighting the role of this. The key ingredient here is organic carbon. In the terrestrial parallel, lignin degrading enzyme actually are known to also create something that looks like a halogenated version of lignin. So there are now two parallel evidence to show that the dynamic could be really interesting in the organic carbon situation. So as a graduate student, that was after the second year of my research, when I tried to grow a lot of phytoplankton, to find out that, oops, it's actually not about the phytoplankton. So I took another detour to now ask two questions. If it's not phytoplankton alone, if they're just there to provide the enzyme, can that be the reactivity of organic carbon? And we know organic carbon as a pool have thousands of known and unknown molecules inside, right? So the easiest experiment to show if it's actually about the compound is to create a single substrate experiment. So you expose individual molecule to the enzyme and see what happens. But we also know that DOC organic matter, they don't exist in the seawater by themselves. So as a grad student, 2 compounds is actually as much as I could do. And I can tell you how exciting it is as a PI now and can look at thousands of them together. But then to look at whether things compete with each other within the DOM pool, I conducted a dual substrate experiment, so mixing 2 organic molecules together. So the first set of experiment, I chose 28 different model compounds that are representative of some major functional group in the organic carbon pool. It was prescribed with rather high carbon concentration, mainly because it tried to suppress the signal from the background seawater. So it's 1,000 micromole carbon was actually incubated with vanadium bromoperoxidase. That is the marine version of the haloperoxidase, activated the reaction with superoxide, right? That is in case that's H2O2. So it was actually incubated in dark so that we don't have any light interference from photolysis and other things for 4 hours. So the reason to choose 4 hours is because this is believed to be a really fast reaction. In the subsequent plots I'm going to show you, there will be a set of data that looks like a gray bar. That will be background production, because whatever that exists in artificial seawater, because the salt was actually absorbed organic from the surrounding, then not entirely organic carbon free, they also produce bromoform. So this plot here shows you bromoform concentration, or the bromoform produced from the incubation of these 28 different molecules representing lignin entities, amino acids, and some organic acids, important as cell metabolite. And what is the really ranking of the production of bromoform? So to orient you, the gray bar once again shows you the seawater being incubated alone, so that you can see the dynamic compared to when you have added organic carbon. There are 3 sets of color here. Red color shows that the compound actually shut down bromoform production. So what that means is the background seawater itself has bromoform, right? But you add this compound that actually shut down the production quite a bit. The black color dot shows you the compounds. Actually, they don't do much in the seawater, so they don't contribute much to bromoform production. And then several of organic acids and 1 humic substance, which is actually an undefined structure substance, are actually enhancing bromoform production compared to the background seawater production. I want to also point out that humic acid is actually not quite seawater soluble. As extracted, it's actually a base soluble acid, so I can only get a little bit in. So that is not really 1,000 micromole carbon, but it still ranks pretty high in terms of the production of bromoform. So this shows you 2 important things. That is actually 3 important things. Number 1 is the molecule reactivity matters in terms of whether they release bromoform and how much. Number 2 is there's something that actually enhance, or this means that they're actually susceptible for releasing bromoform as organic molecule. But the very interesting part is, why do they shut down bromoform production? And one thing to come in quickly into the mind is, like, do they then inhibit the enzyme? I did a abiotic parallel study to just directly use bleach instead of enzyme, and to show the same result, showing that it's actually not a enzyme inhibition reaction. And that actually gets pretty exciting. I'll tell you why later. So amino acid compound and phenolic compound prohibits carbohydrate, which is basically most of the organic carbon pool actually do not do much. Important cell metabolite and humic substance enhance bromoform production. And then in the dual substrate experiment, I chose urea, which ranked the first in enhancing bromoform production, and mixed that with 3 of the inhibitors and to see what would be happening. And then use 1 of the compound, D-glucose, which did nothing in bromoform production, as a reference to see, in that case, what does it really mean? And then a control really means adds nothing, but ultra high purity water. So the mixing ratio is as follows. So is I added like 50% inhibitor or this other group of compound, 10%, 0.5%, and 0% in relative to urea, and then redo this incubation experiment to see what is going on. So on the left-hand side here shows you tryptophan, vanillin, and phenol red, 3 inhibitor being mixed with the enhancer urea. Once again, the gray bar shows you background seawater production. It's striking to show that at 50 and 10% mixing of the inhibitor, they still entirely shut down bromoform production. Even if they were mixed with very low ratio, 0.5%, they still inhibit the production quite a bit. So in the last bar here, 0%, which means entirely urea, of course, the production become normal, as in how you would expect from urea. And then the D-glucose and ultra high purity water incubation, what it shows you here is just a simple dilution of urea, confirming D-glucose indeed did nothing, and it's actually similar when you compare to when it was mixed with nothing. So then the question I want to ask is why do aromatic compounds, phenolic compound, inhibit bromoform production? Phenol red is in fact one of the substrate that you can use to assay bromoperoxidase activity, and I intentionally choose that as one of the substrate because I know that it wouldn't do nothing, right? It clearly is an assay thing, so something had happened to that. So that means this inhibiting something is leading to something else rather interesting. So with bromoperoxidase, phenol red will get brominated, in this case, polybrominated into bromophenol blue. When I was a grad student, I didn't have a ultra high resolution mass spec, so I did what I could, which is an absorbance study. So initial, you can see bromophenol blue, this mostly is actually background scattering. But then once you activated the enzyme, you can see the concentration of bromophenol blue substantially increased. So this shows that while phenol red as a model compound inhibit bromoform production, it actually turned into bromophenol blue. So then what would be the case from one of the lignin phenols? This is actually a fluorescence study. Once again, didn't have an ultra high resolution mass spec. So to try to see did halogenation somehow change optical property of a organic compound. So gray bar here shows you the control, which is with the enzyme, but not activated with HO202. The black data here shows you enzyme being activated. So you can see the moment the enzyme being activated, you have a substantial shift in the fluorescence signal of this ferulic acid, which is one of the entity of the lignin phenol. So that tells us not only when they inhibit bromoform production, something happened to them, something actually structurally happened to them that changed their chemical characteristic. Then one question to ask ourselves is, okay, so what? So what is like, what does that mean? Going back to the parallel that I drew for persistent organic pollutants, once again, because they have aromatic structure, we ask, then are we somehow making refractory organic carbon when we are not releasing the ozone depleting substance? So one of my colleagues actually has this study conducted with X-ray adsorption near edge structure spectroscopy. So this is a schematic that she basically shows either through a Fenton-like reaction or bromoperoxidase reaction in the ocean, different organic molecules can be brominated. Some of those can actually be partitioned to the particular organic carbon and may actually be able to export it from the surface of the ocean and persist in the deeper ocean or even in sediments. One of her data shows on the left-hand side here, shows you the square solid line, the bromine that is bound to a organic carbon. And the dotted line here shows you the organic carbon concentration at the depth of Bering Sea sediment. This is not age dated, but suffice to say, you can see that we actually have a decoupling between the organic carbon and organic bromine, but then we also basically have the evidence to show that we found that in marine sediment, hypothetically being exported from the surface ocean. The decoupling here is rather interesting, but we also have another elevation of bromine in it, organic compound. So then it leads to the question of, are they really stable? Who are they? Because Zhang's method do not tell you the chemical characteristic. So at this point, we have evidence that non-volatile organic carbon that are naturally produced exists quite a bit. If we go back to this figure here, it's actually at ppm level, which is not negligible. The problem is, at this point, we still don't know how HOC affect the cycling of carbon, mainly because we still really don't know what they are structurally and what does it mean biogeochemically. This is because historically, we have a hard time characterizing what they are because mass spectrometry limitation and resolution of mass spec. But with the advent of ultra-high resolution mass spectrometry and new algorithm, we are now in an exciting moment that we can start to unpack what they might be. So in PNNL, I was fortunate to get funded to develop an algorithm processing ultra-high resolution mass spec data. So this is the 21 Tesla FTICR PNNL, and this is a QTOF in my lab. Subsequent plots and results would be coming from these two platforms. So ultra-high resolution mass spectrometry, if we think about DOC as one thing as a black box, then it means the ultra-high resolution start to kind of unpack them into a bunch of stakes or peaks that we call them features, because until we identify them, we really can't say that is a compound, right? So the idea is within one nominal mass, it can resolve many different peaks accurately that enable formula assignment. While this still don't give you structural information, but with the formula assignment, you can start to at least understand what kind of element do we have in a compound. And based on the carbon and hydrogen number, we can start to at least understand, do we have a saturated, unsaturated carbon, or are they mostly possibly aromatic, for example? So Liz Kuzhwinski, who I did postdoc with at Hui, has a algorithm that can assign identity of these peaks formulas based on normal elements, CHONP The problem is, even with ultra high resolution, the more element you allow, the more false identity you will have. So then if you start to throw in halogen without further consideration, what does it mean, right? So the grant I had funded actually developed then this algorithm to basically is a pattern recognition algorithm. We have millions of theoretical computed formula that we would then halogenate them. Because the unique natural abundance of halogen, in this case mostly chlorine and bromine, a originally non-halogen compound have one major peak, and the C13 peak is really low in a high resolution mass spec in a complex matrix. This peak sometimes is not visible. So usually you just see one big peak. As it starts to get halogenated, you start to get very diagnostic isotope pattern. So we can use that along with exact mass to confirm, at least, is that true that this compound has a halogen on them? So using this method, I induce a laboratory control halogenation and shoot them into the FTICR. And I must tell you, this was one of the very exciting moments because I started with 2 compounds, right? So now I get to deal with thousands of them, which is sometimes it's actually not. This is also it's different kind of problem. So this lab control halogenation, I use Suwannee River natural organic matter isolate and dissolve that into allotropic seawater. So to give it some more or less well understood DOM isolate, and then to induce halogenation. And these are the outcomes. So for those of you who are not familiar with this kind of Van Creden diagram, each dot on the plot here shows one chemical feature that has a chemical formula being assigned. It's being projected into 2 dimensions. In the x-axis is oxygen to carbon ratio, and the y-axis typically is H2C, hydrogen carbon ratio, but because halogen actually substitute hydrogen, so in this case it's really X2C, X now being hydrogen, chlorine and bromine. I did not look at iodine mainly because iodine do not have a very substantial natural isotope abundance, so I was not comfortable saying that I saw that. So the take-home message through this incubation, so I actually incubated beyond 4 hours as I did as a PhD, so it did up to 48 hours. One thing I would like to highlight is that number one, this control, basically 0 hour, means nothing has been halogenated intentionally yet. Has halogen organic compound existed inside to begin with? This can come from some of the organic matter from seawater or Suwannee River. So you can see not only that we have HOC, but we have quite a bit of HOC. However, as we start to intentionally induce the halogenation, something interesting happened. You can start to see this blob of HOC start to expand to more oxygen-rich direction. So what that really means is actually to be kind of unpacked in the subsequent talk, but using Van Creden, sometimes you can draw domains and kind of see, okay, what are those compounds? And I must tell you that please take this kind of classification as a grain of salt. I'm usually not a fan of this kind of domains because it don't necessarily mean what they are. But some of this, like tannin-like, so suffice to say, some of those have phenolic features. Especially if we believe this is indeed tannin-like features, the growth of HOC as the induced halogenation happened extended into this region. So knowing how the single substrate experiment happening and now comparing this complex matrix, I do have a reason to believe this might be real, as in phenolic structure is really susceptible for halogenation, and they did not release the volatile ozone depleting substance and stay as halogenated phenolic compound, lignin-like compound, whatever, how we define that. So the lab-induced experiment shows that, okay, I literally induce a halogenation, I see more of those, so the assignment I can more or less trust, right? So then the next case study using the FTICR is to look at some preserved marine sediment and see, okay, so we induce that, we see them, but what if in the real environment, do they exist? So my collaborator at PNNL has multiple cores at the Puget Sound that dates back to actually pre-industrial time. This is actually dated based on, I think lead isotope. But today I'm going to show you this particular core PS1, that was where the star located. There were background information being collected at this core that are somehow related to halogen organic compound. So for example, total PBDE, polybrominated diphenyl ether, which is one of the flame retardant that sometimes older people will see that in their home's furniture to prevent fire happen. As you can see, this is an anthropogenic compound. I guess it was invented or at least found in Puget Sound after the 60s, peaked somewhere in the 90s. And then I guess as the phase out start to degrade it. So this marks one of the anthropogenic signal. The sum of lignin and pyrolysis polyaromatic hydrocarbons. This came from combustion, I guess, wildfire and things like that. And this would then shows us terrestrial material discharged to the Puget Sound. So we can see events of the peaks actually are offset, so marks the height of release of brominated anthropogenic carbon. Some of the lignin release peaks later in the 60s, and then aromatic compounds that are not phenolic has a slightly earlier peak. Then to see, okay, can we see any halogen organic compound? Are they related to this background information that we collected? So I was rather excited to find a bunch of halogen and organic formulas. At this point, please understand FTICR is not quantitative at all. So this signal, they are relative abundant. So basically calculated the sum of any formula, the sum signal from any organic compound that being assigned carry a halogen divided by the total signal of everything and calculate percentage. Similarly, this organic bromine signal is anything that being assigned as bromine, I sum the intensity from the mass spec divided by the total feature intensity. So this more or less tell you qualitatively, do we have elevated signal of organochlorine and organobromine? And what is exciting here is that the highest elevated peak of organochlorine and organobromine, they were pre-industrial, shows us that those are natural. But then we have all these events, right? We have the maximum of PBDE marks the height of the anthropogenic input, and then lignin input and combustion input through nature. Then one question would be, number one, this would be natural. They were preserved now at this point, at least for hundreds of years. Are they considered stable? Question number one. Number two is, are these peaks observed around 1995 where PBDE were peak? Are they then natural or anthropogenic? It's probably hard to tell, but I tried, right? So showing this in Van Keren diagram again, I observed in this particular layer, 1995, more than 4,000 chemical features being assigned, projected on this Van Keren diagram. 99 chemical features were being assigned as HOC. So in slide number one, when I told you in the natural product world, they identify 5,000 through the decades, 99 for one sample is kind of exciting. So I was like, oh, wow. So that's important. And the star sign shows brominated compound because I want to know, are they related to the PBDE compounds? So while formula assignment cannot give you a structure, based on the formula itself, you can more or less put together possible candidate of structure. They are not necessarily the confirmed structure, but at least based on the saturation of the compound, you can start to understand, well, they may at least truly have a ring with that. So these are the possible structures. One of these looked like probably part of the PBDE compound. Some of this look like maybe half a PBDE compound. So one hypothesis would be, okay, so the signal that were elevated also through the FTICR that is in the 1995 layer where the PBDE was elevated, could they be actually metabolic products or partially degraded product of PBDE? So that's one hypothesis. So the next thing is then we can isolate those formulas and plot, once again, relative signal through time and ask ourselves then, okay, so what does that mean? Now, this is actually exciting, particularly for this that profile that I'm seeing here. They were found in the 1995 layer that I thought it might be half a PBDE, but they peak pre-industrial time. So going back to the lignin bromination hypothesis, some of this could be actually natural products and probably from halogenation of lignin-like structures. That's one of the hypotheses. So try to understand the lignin dynamic. Lignin dynamic, we can use some of the parameter here. I won't go too deep into the lignin biomarker, but suffice to say there are two ratios, C to V, S to V, which is some of cinnamol group, syringil group ratio to vanille group. This ratio tells us the shift or the sourcing of organic carbon. So if we have a higher C to V ratio, it shows the shift from woody to non-woody substance. If we have higher S to V, then we are shifting from gymnosperm to angiosperm. So if we then look at the ratio of lignin biomarker, we can see also some pretty interesting trend happening before this elevation. This is here, and compared to the elevated C to V and S to V, that shows us somewhere in, I guess, the early 1900s, there is a ecosystem or vegetation input change. So this shifting is interesting because they are significantly correlated with the two formula identified being brominated compound having this possible structure. So for a environmental data that are dated back to hundreds of years ago, I must say that I was pretty impressed with the correlation here. So then can we just hypothesize that at least some of the brominated compound, particularly so as an example here, these two brominated formula may be related to lignin biogeochemistry. Again, confirming the possible link of halogenation to organic carbon cycling. So I hypothesize that non-volatile HOC is real, and I show that it was found. Then the question is, can they be preserved for much longer than 100 years? So, of course, now we're shifting ecosystem to terrestrial environment, but this is the only ancient product that I can get. So, but this is actually another very interesting opportunity. So these two cores I'll be showing you collected from southern peatlands, tips of South America. The reason to think about peatlands and carbon dynamic is because they're significant carbon reservoir, number one. Number two is for unknown reasons. Some scientists actually found high levels of organohalides accumulated in peatlands, although they don't know what they really are structurally. So this great opportunity collaborating with a faculty member at Texas A&M, Julie Loisel, we have two cores that we examine. The HB core is actually at this channel here, is actually what we call a coastal core, so exposed to seawater, at least in current day. And the pan core is slightly more inland. So this can help us to understand the hypothesis of, do more bromine or halogen exposure increase the number of halogen organic compound? So the two cores were dated back to thousands to 10,000 years before present. So this is a great opportunity to also show the Zhang's data, because this is the first time the mass spec and Zhang's were parallel. I would just want to say Zhang's, once again, while it's actually not structurally specific, it tells us for sure that a bromine is indeed bound to a type of organic compound. So the HB core are in the blue symbol, and the pan core data in organobromine is in the maroon dots. What is fascinating is that we see a accumulation of organobromine in the HB core, which is the coastal core. So the correlation between the age and the concentration of organobromine is actually rather significant. It's actually with the Spearman rank correlation of 0.73. Now, when it goes to pan, which has actually dates way earlier in before present time, the dynamics start to look slightly different. So we still see in the 1,000-year timescale accumulation of organobromine. If we have to fit some fit line, I would say we probably stay pretty stable through thousands of years, and then you have a decline. Possibly that is either a debromination happening when the sediment is going anoxic, the peat core going anoxic, or thinking about 10,000 years ago, we actually may have different sea level things. So this decline, at this point, cannot safely say what's going on. But at least we know from a 1,000-time-year scale is actually, we are seeing organohalogen. Zincs, however, do not tell us molecular level information. So we use solvent extraction. Now this is one thing to be clarified here. Zincs are done directly on the sediment themselves, so the solid phase. We cannot shoot the solid into the mass spec, so we have to solvent extract them. So there might be some decoupling there, just as a caution. So we run that in my QTOF in my lab and found more than 150 possible HOC candidates, and this time mostly chlorinated because, I guess because they are on land. But we do find some brominated candidate. So the three formula that I identify that I want to show as a highlight because they actually have suggested structure. If we look at all three of them, the commonality is they have a aromatic core similar to the hypothesis way back when I was a grad student. For this particular compound here, we have a peak somewhere in the 100-year level, but that totally did not follow the zinc data, but that is zinc brominated compound, is chlorinated. But then it shows actually biogeochemically, there's not just simply just accumulating. Within molecular level, there's much more dynamic going on, only seeing this three example structure. And actually, this compound here, polychlorinated, has two peaks. Actually, now you can see signal in this pan core. So this one only found in HB, this one only found in pan, different dynamics. This actually is a suggested structure correlated to a soil algae product. So they may not be necessarily really halogenating the material. It could actually be the metabolite from some of the organism to begin with, but they are still halogenated. Now, this brominated compound is one of the formula that we have signaled from both cores, and it's interesting. So, number one, this has a peak at closer to the 10,000-year age, although why they're being preserved, it could be they're actually that stable, or it could be somehow they're being protected from other layers of material piling, or there's something ecologically interesting happening to produce this compound. But in the HB sample, we can see a clear accumulation through time in before basically within few hundred years of time scale. So what I hope to convince you at this point is that when we have parts per trillion level of volatile organic HOC being produced, beneath that is linking ppm level organic carbon that is being affected. And I demonstrate that in marine sediment and also found them in the peatlands work. The peatlands work is still ongoing, so there's a lot more to talk about what is the biogeochemical role. And then the next step is, of course, look deeper into mechanistic formations. I'm fortunate enough to be funded by NSF to look further into, number one, what is formation mechanism besides just the enzyme, or if there's enzyme, why and how, like why do phytoplankton upregulate that enzyme to begin with, and try to understand, do we see then HOC in deeper aged seawater to basically link the whole organic carbon cycle, hopefully, to kind of shed some light on that, not to necessarily say that I solved the problem. So this is actually a work starting from grad school, lingering through postdoc, and back to my professorship at Texas A&M. So it's super exciting, and this cannot be, done without all the funding agencies and hardworking grad students and postdocs and scientists in my lab. So with that, I think I can stop and open the floor for questions. Thank you so much.
If there's any questions, go ahead. Put up your hand or Eric, yeah, you want to go first?
So, what, does the enzyme exhibit a strong preference for chlorine versus bromine, and do you see that preference in the products that you make?
So the enzyme itself, the marine version of bromoperoxidase, it only hypothetically has the capability to brominate and iodinate. So with iodine being lower in concentration, so even the iodination happened faster, somehow the brominated product dominate in the marine situation. In the land side, the chloroperoxidase actually can halogenate all three halogens, but the kinetic is still faster in the brominated compound. But somehow, I think on land, the chlorinated version of organic compound is more frequently found, although the enzyme kinetic really didn't work that way. So there's a kind of a bit more to think about why that was the case.
But they do have the chloroperoxidase in seawater, or no?
Not in seawater, hypothetically. Not to be suggested today, but there's the belief that the bromoperoxidase is dominant. There's a marine version of chloroperoxidase is the land version.
Yeah, question about the, first, thanks for a really nice, clear talk. I didn't know anything about this subject before, now I'm interested. When you were showing the depth profiles of the zanes, that was showing you actually had like a greater proportion of these bromine and chlorine containing compounds, maybe not in the food, but in seawater samples, it was clear that you had higher values deeper. And was that just that you're plotting the proportion? So
This not this part.
You're losing other things at the top, and that gives you a greater proportion of the chlorine containing and bromine containing things deeper. But then if you compare that to your single compounds, it looked like those patterns were similar. So
yeah, so this particular one, this is actually not Zhang's, this is FTICR. So I guess this is the reason. However, in the peat example, for example, we asked, okay, if organobromine seems to be accumulating, then the question is, how does it relate to organic carbon? If the carbon is actually constant, that shows us maybe increasing bromination is happening. But if the organic carbon is actually decreasing while bromination is increasing, that would suggest a preferential preservation of organobromine. At this point, I still need to kind of have some firmer evidence to say one way or the other.
okay.
Yeah.
So I guess the other question would be, is there nothing that degrades these compounds?
Yeah, so the natural product is actually not as well known for, if we draw the parallel for contaminants, the degradation actually is more efficient in anaerobic situation because you can do reductive dehalogenation. And when you dehalogenate, you're basically now making the carbon accessible. Otherwise, you can only just attack the carbon by oxidizing it. So I guess it's quite a bit of, I guess, oxic and redox situation that need to be considered as well.
Yeah, if this was the long-term transit.
The Zangs?
Yes. So you have those 2 points in the end, general outliers.
These two, yeah.
Can you talk about that, because it's the same range as potentially the delineation?
Yeah, so I will actually have a meeting with my collaborators next week to talk about this data. So it's actually super interesting because it may not entirely because of the degradation, it could be actual deglaciation or sea level change during those years. Because I think at least at this mark we have a sea level change situation. So I cannot comment on what is the geological reason for that, but that's a great question. It's actually should be investigated in terms of along with ecosystem dynamic.
And with the ones in the middle, like those.
This one?
Yeah, and the lower one.
This one? Yeah, I'm not sure at this point, but I can follow up when I find out more.
And then, since you talked about the anaerobic situation, isn't the whole or mostly anaerobic in a situation like this most of the time? In order to allow for the carbon to accumulate.
Yeah, yes. However, I guess if the kinetic is slow, then maybe they get to be preserved for a while, and then so that whole thing applying to natural product is actually not entirely known. So I think it could be dehalogenation purely because of this data, but I cannot say this actual mechanism because it could be something else, right? Because if, like you said, if this whole thing is anaerobic and if the top look seems to be more or less stable, then this change may mean entirely something else. So before I flew out here and look at the sea level, and I'm trying to see, do we have a receding or increasing sea level? Is some sort of event is happening globally around this time, although I don't know specific of this location. Yes.
So in your earlier slides, when you talked about compounds that promote qubit hallucination, I was a little surprised to see urea in that mix. So urea hydrolyzed is two, and the carbon, right? So why do you think urea is that? Or is there like a lot more going on than?
I was also rather surprised about that as well. I thought it may be my mistake, so I repeated that experiment more than 10 times to make sure that is actually real. And back then I couldn't understand why. And then when I talked to a civil engineer who deal with bleach and urea stuff a lot, apparently it has something to do with the nitrogen situation that is not necessarily entirely due to the molecule itself, but other, I guess, something dynamic about nitrogen happening. But mechanistically, if you look at the structure and halogenation, I have no clue why that would be the case for urea specifically. I think I discussed that in my paper. I need to look back. Sorry, that was more than 10 years ago. I'll get back to you though.
Are there sterile these samples?
The incubation? It is as sterile as it can be, as in I autoclave the water before I do anything in 0.2-micron filter.
Okay, so you're not stimulating like how it works.
No. The idea is to try to isolate, to make sure this is actually an enzymatic thing and not other intracellular production. Although there could be other compound like methyl bromide that is actually facilitated by an entirely different set of enzymes that could be intracellular. But for this particular set of compound, it's only enzymatic and exocellular. Do we have any question from?
I didn't see any from online.
Okay.
If there's any other questions, and let's thank. Oh, you have one?
One quick one. I mean, it seems to, what you seem to be saying is that these new organobrominated compounds that were spreading all over the environment are actually relatively unimportant compared to these natural compounds in terms of their abundance, and that would seem to be also these people.
Yeah, from a biogeochemical sense, I guess that would be the case. For health risk assessment, that may not be the case.
Right. Okay, that's what I wanted.
Yeah.
All right, let's thank you now.
Thank you.